Hello, everyone. Can you hear us?
Yeah.
Perfect. All right. So thanks for joining us today. We're going to be doing a revision session for you for Silesia. I'm Osman, and this is May next to me here.
Hello.
So May will be guiding you guys. Oh, we have one more person. Oh, hello, YM. Allowed to talk. So we've allowed you guys all to talk. So we've allowed you all to talk. So if you have any questions, then feel free to send a text on the messaging forum, or on the Q&A, or on the chat, or just to raise your hand as well. You can raise your hand, I believe, or you can just unmute yourself and ask a question. So May will be going through some topics for you to try and show you how things are done and how to expect the exam to be like. May is currently a fourth-year dental medicine student. She's successfully finished 4 years already. And as you know, in dental medicine and medicine is roughly the same in the first 3 years. So medicine and dentistry students study exactly the same subjects in the first 3 years. So she's been through a similar experience with what you're about to go through. Oh, hello, we have another person joining as well. Okay. So May will be going through some topics as an example to show you what it's going to be like to have these lessons. And it's up to you to choose if you believe that you're going to benefit from them, or if you see value in them. We'd love to do more sessions with you. So this would be a free taster session to try and give you as much insight as possible in order for you to succeed on the exam. So I'll be allowing May to take over from here, and we'll start the lesson. Does anyone have any questions before we start?
Yes, I have a question. Hi, Osman.
Yeah, hi, Federico.
Hi. Do you also tell us the conditions? For me, it's not clear how it will be. How deep we have to go to the matter in the exam. That's a bit my question. Yeah, it doesn't seem so much time we have to talk. So.
Okay, so May will go through this and give you some insight. Now, generally in Eastern Europe, how it is you will pick a topic from the list of topics that you're given. And then when you pick a random number, it will be assigned to a topic. For example, eukaryotes, let's say. And then once you've done that, the teacher will pick a paper off the table and it will say eukaryotes for, let's say, number 7, because you picked number 7. It will be an oral exam like this one that we're having now, a discussion where on video you're in front of us. And May will give you more information about this as she goes along. So don't worry, she's here, she will help you. And if you have any questions, we're always here to help you and give you as much guidance as possible. We want you to succeed in this exam because it's one of the toughest exams always. Getting into medical schools is the toughest part. So we want you to succeed with it. We want you to study hard for it. And we want you to be as thorough as possible with your knowledge, because answering a question when you get asked in the exam with 1 or 2 sentences is usually not so good. So you want to answer in as much detail as possible. You want to give so much details. You want to impress the examiner. You want the examiner to be like, wow, this person is really knowledgeable. They have prepared. They have tried their best, because you've all tried in your home countries. Everyone here, I believe there's several people from all over Europe. You've all tried from your countries, and it's not easy getting into medical school. And it's not easy as well abroad. But you have our support and you have our help. And you have to do your part in order to make sure that you're fully prepared for it. We're here to support you as much as we can, but you also have to do your part. All right?
Yes.
Any other questions? Okay. So we have here a few students with us. Thanks for joining us, everyone. I'll be giving the floor to May, who will be taking over and sharing her presentation with you. So you can all, this one? The screen. It will share. Okay.
That's fine. No problem.
Share.
Yeah.
Here we go.
And then if you just go to my slide, just yeah.
Is it on
Then
Google Drive?
the arrow. There we go.
Yep. Cool.
And
Okay.
press present.
All right. It's okay.
Okay, hello, good evening, everyone. So thank you very much for joining us. So today, as you all know, we will be talking about getting into Silesia University, so Poland. And we will be talking about the logistics of the exam, so how the exam is laid out, how it is structured. We will talk about the requirements of each question, and we will talk about how we can really ace or master the exam questions to get you fully prepared. So sorry, I'm just plugging in my earphones.
Okay.
Okay.
Try it now.
There we go. Can you hear me slightly better now?
Yes. Yes. Also before, I mean, it was okay
Also
to me.
before was okay?
Yeah. Yeah.
Okay.
That's good.
It's a little bit better now because the audio is slightly clearer. Okay, so without further ado, for your lovely introduction, we're going to begin. So, okay, thank you. So, how can we master the Silesia exam? So this is a question that many ask us. If you have already seen the exam mark scheme, they are full of questions in biology, in chemistry, in physics, and sometimes it can be very overwhelming. You know, how do I have to structure my answer? How much time do I have in the exam? One is bombarded by so many questions. So after today, hopefully you will know a clear structure of how the exam will be laid out, and hopefully it will be easier to attempt each question. So sometimes all students at one time in their life, they get so overwhelmed by so much information. How do we manage time? How do we cover all the topics, especially in fourth year as well? It gets harder as you go into university. And sometimes one has just got to stop and think and say, okay, how do I manage and how do I fit everything into the schedule? So a famous quotation from Albert Einstein I like to choose. You have to learn the rules of the game and then you have to play better than anyone else. So I think this is very relevant in this case because there will be so many candidates, so many students who will be applying to Silesia University, and you want to stand out as a candidate. You know, you want to stand out as a student, and you want to show to the examiner that you know all the details for that specific subject that they're asking you, and you just want to impress the examiner. You want to give them a lasting impression, let's say. So sometimes the students are like this, but one thing we have to remember is that you never fail until you stop trying. Again, from Albert Einstein. Okay, so as Federico asked before, that was a very good question. So we're going to go over the exam format. So how is the exam laid out? So especially during these circumstances, during this Corona time, the exam will be held online. The number of topics that you will choose is one topic chosen from 2 subjects. What does this mean? So we know on the mark scheme, there are 3 subjects. There's chemistry, there's biology, and there's physics. And the most popular choice is chemistry and biology. Some people choose physics. I would recommend to choose biology and chemistry because there are a varied number of topics one can choose from. And the examiner will say, okay, your topic is chemistry, your topic is biology. Can you randomly choose a subtopic? Then you choose it, and then you have 15 minutes to write your answer. Now, this 15 minutes is very, very crucial because it is the time where you can structure your answer and a time to put all the little details in that you know relevant to the question. And so after the 15 minutes is up, the examiner will turn to you and say, can you please tell me about, for example, the structure and function of the heart? And so you will use your written notes as an aid for yourself. But usually the exam wouldn't be online, and you would actually give your written answer to the examiner. So just think the exam is going to be held like that because it will encourage you to put as much information down. Some students, they think of this 15 minutes of just writing 1 or 2 words down or a few sentences, but it's a waste of time. I would rather encourage you to write as much as you know, structure your answer, and then you can use it as an aid when talking to the examiner. And then the total duration of the oral exam is around 30 to 45 minutes, depends on individuals. So far, do we have any questions? Anything you would like to ask me?
Yes, I have a question. You said so. As I understood, we have one. So we can say, for example, me, for example, would like to take biology and physics, and so it is then at the end the examiner who will decide if I will have biology or physics, right? And then about the subtopic, I didn't understand well. I mean, I cannot choose the subtopic, right? He is the one who will tell me choose a number.
Exactly.
Okay.
Yes, you are correct. So basically, as you said correctly, there are 3 subjects: is biology, physics, and chemistry. So before going to the exam, already have clear in your mind which 2 subjects you want to choose. So in your case, you would like to choose biology and physics. Is that correct?
Yeah, I mean,
Yes.
I'm thinking about chemistry now because you recommended it, but I don't know. I like more physics than chemistry, but okay.
Oh no, it's up to you. It's personal choice. It's just because chemistry has a larger number of topics compared to physics. But if you're more passionate about physics, go for physics. And in regards to the subtopics, sorry if I made that slightly unclear. I meant so each subject, chemistry, biology, physics, has a number of points, has a number of questions. So as you said, they will say choose a number from 1 to 60, let's say for biology. And if you choose number 7, they'll read out the question and then you start writing like that.
Okay, this is meant by the subtopics. Okay,
yeah,
I didn't.
Subtopics.
Okay,
yes.
Thank you.
No problem. Any other questions?
Really clear?
I'll just have a look. Oh, we've got 7 participants. I'll just put allowed for a second. There we go. Who was talking before? Was that Mario? Did you raise your hand before? Someone raised their hand. I'm not sure who it was. It was here. Allowed to talk. Yeah, yes. Okay.
Yeah, hello?
Hello.
Hi, I wanted to know the exam is proctored or not.
The exam structure?
Proctored.
Sorry, can you repeat that again?
Proctored. Proctored, you know, proctored exam? Proctur. Proctor. You know what I mean?
No, I'm unsure what you mean. What do you mean by that?
Proctored, it means that there would be like a proctoring company, like for example, Procter U or something that, let's say there would like be somebody who is watching me doing the exam and watching my screen?
Right.
I'm just wondering.
No, in regards to that, I'm not aware of that. If someone else is observing, I think it will just be you and the examiner. It will just be like that. The examiner is from the university and yourself. There won't be someone
see.
else next to you, or no.
And what about the exam duration? How long would it be?
Yeah, so in total, you've got that 15 minutes for the preparation of your written answer for the oral exam. And then you have, for the actual oral exam, 30 to 45 minutes.
So it's
let's
all
say
like around an hour,
around an hour, yes.
the whole entrance exam.
The
First,
whole entrance exam, yes. It
first
could be
15
slightly
minutes?
less as well. Yes, the first 15 minutes is for
Are-
that is dedicated solely for that preparation. So probably you'll go into the exam, you'll say hello to the examiner. They'll
I
say,
see.
can you please pick your topics?
I pick one, and then I say one number, and then I just answer one question for 15 minutes. I just keep writing it down. Then we move on to the actual oral section. Is that right?
Yes, but one topic chosen at random from 2 subjects. So you'll choose one topic from biology and one topic from chemistry, for example. Or
I choose my,
one
I could,
topic. Or
I could, for example, say circularity system in biology, and then in chemistry, I could say, I don't know, pH level, something like that. Is that what you mean?
exactly. Yeah, that's what I mean. But you will have to choose a number. Rather
so
than
it's
saying the
random.
topic, it's completely random.
I see.
For example, they'll say, can you please choose at random a topic in biology? And you'll say number 25 in biology. And can you choose a topic in chemistry? Number 27. And then they'll say the topics to you, and then you have to start writing it down.
Would I need a web camera? Do I need like an actual computer, or can it be done with my laptop, or are you
No,
unaware?
laptop is fine. I would recommend laptop, computer, iPad. I wouldn't recommend phone.
All right, great.
Logistic wise, it doesn't work, but it would be
Yeah,
better.
of course.
It would be better on laptop, computer.
Yeah, laptop is
And
completely
also,
fine.
actually, if you have earphones or if you have a headpiece, it will help you. With
to hear.
the audio
So
quality.
the audio would be clear. Yeah, of course.
Exactly.
Right. Thank you. Thanks so much.
No problem. Is there anyone else who has any other questions?
Sorry, just one question before we continue.
Of course.
So is the examiner going to ask us more than one questions for each topic?
Okay, so do you mean, for example, if you choose at random eukaryotes and prokaryotes?
Yes.
And in the oral exam, they may refer to another topic. Is that what you mean, like cross-linking? So they may start to overlap onto another question. Is that what you mean?
Yes.
It could happen. It could happen. But the topic that you have chosen, for example, if I pick at random, I choose the structure and the function of the heart, they will probably ask you to talk. They may ask you a few questions just to clarify a few things if they think that you're unsure or if you have lacked detail in your answer. But probably the questions they will ask will just be primarily on the question you have chosen. There may be some cross-linking, but they will mark you on the answer that you have given for that specific question.
Yeah, thank you very much. I'm asking because some of the topics are a little bit general, and there are quite a few information that you can give the examiner.
Yes, that's actually a very, very good point. And what we'll do is, after a few slides, like during the presentation, I will give you a few sort of sample questions and how to structure those specific questions. Because some, as you said, it's hard to put a lot of information. So I will tell you how to approach those type of questions.
Okay, thank you very much.
No problem. I saw some hands being raised. You can go ahead and ask your question if you wish.
Yes, I mean, it's again me. I think it goes in the same direction. I mean, yeah, because I read that it's high school level, of the knowledge, but I've seen also on the transcript which Medlink sent us, you know, with the answers for the questions. There are
Yes.
just like answers which are really short and which I couldn't, I mean, which would be too short to talk about 15 minutes. I mean, I would have that in 1 minute perhaps. You know,
Yeah.
it's just not so much how do you say stuff.
Not so much detail.
Yeah, information, and that's a bit for me difficult to value. How much do I have to know about the topic to fill these 15 minutes, and at the same time, it has to be high school level, so it doesn't go like our biology or chemistry and in the university, because I'm transfer student. It's a bit for me too difficult to understand how deep.
Okay, all right. So we do say high school level, but the more detail you can give in your answers, the better. I think this is the advice that I would give you. The mark scheme that we gave to the students that you have received, it was made from a group of students here at Medlink. But this is just, let's say, a basis, just a foundation. And I would recommend you to use it as like a skeleton. But these courses that we're going to be doing, like these little intensive courses. So for example, today will just be a taster. We'll add more flesh to the bones, or more flesh to the skeleton, because more details have to be given. But for example, if you are a student which is transferring, you could add more knowledge, like prior knowledge that you know from your chemistry subjects or from your biology. The more knowledge, the more you impress the examiner. This is my motto.
Okay. Yes, because there are topics like, I don't know, the Krebs cycle, and in the Krebs cycle, I mean, if you had already biochemistry, you could just tell so many things, but
Exactly.
on the same time, you have only 2 months or 1.5 months to prepare for the exam. So it's a bit, yeah. Okay, thank you.
This is why we are here to help you. We are here to try to let's say, funnel the knowledge in a way or in a structure where we can, you know, give it to the examiner. So rather than just writing everything we know not in a specific structure, trying to channel that knowledge in an order. So when the examiner asks us, okay, tell me about the structure and function of the heart, for example, and this is one of the sample questions that I've chosen to go over today, we will go in a specific sort of order. So first of all, we would say, okay, the heart, which system does it belong to? Second of all, after we have described that, we would describe the borders and the surfaces of the heart. Then we would describe the function of the heart. Then we would describe the function. Now, I understand your question because, for example, with the heart question, it says just structure and function. It's so vague, and you can speak about so many things. So I would advise structure, microscopic structure, and gross structure. As well as that, how does the structure and the function, how are they related to one another? And then you can add more details at the end if you have time. So it's just trying to take the examiner on a journey and showcase what you know. That's the whole aim of it. But again, I emphasize, the more you put, the better.
Okay, but they aren't considering like, okay, the student is now transfer student, so I will expect more from somebody else who is.
They may.
Oh,
because
okay.
you are a transfer student. They may. So they will expect you to have a slightly higher level of knowledge. But again, try to attempt to structure your answer in a specific way rather than putting everything that you know. Try again to give them a structure. But yes. Even though we do say higher level knowledge, it will vary between those students who are just coming from university, transfer students, and those students who are just coming from school. But I mean, if you have any other questions, you can still ask me, and the topics that we'll look at today. and we can see how we will approach them.
Okay, yeah, I just hope it won't be so much detail, because if I have to study it how I had it in 2 years, then I would, yeah, it would be too detailed. So yeah.
It's almost like giving them a snippet of what you know. Okay. Like they will know that you are a transfer student, but they won't expect you, as you said, the time you have to cover two topics from each subject. You won't have time to go through diseases of the heart, through you know, the nitty-gritties of the subject. You'll just be able to give them, you know, a good consolidated answer.
Okay.
Okay,
Thank you.
okay. Is there any, can I help with anything else? Any other questions?
Can you hear me?
Yes, I can hear you. Hello.
All right, perfect. I have one last question. Is it like what's it called? Can I like be seen through the camera in the exam or no?
It will be like this. So if you pretend I'm the examiner.
Oh, just like a Zoom meeting.
Like a Zoom meeting, exactly.
Oh, all right.
It may be on Zoom, it may be on Skype. I'm not sure about that. I'll ask one of my colleagues specifically which is used, which platform is used. But yes, it will be like this. They'll be able to see you.
Okay, great.
I would recommend to put your video on. That's very important.
What?
I would recommend you to put your video on rather than just speaking on audio.
All right, I'm just wondering, you know. All right, thank you.
Okay, Duki, I'm going to move to the next slide. But if you have any other questions that crop up, raise your hand, say, oh, can I butt in? No problem, you can interrupt me. This is why I'm here. Okay, so let me just move to the next slide. Okay, so now I come on to the structure. So all the questions that you asked me this evening, they are very, very, very good questions. And I will try to cover as much as I can. So first of all, how do you structure the answer? So what is very important is that we should identify and define the key words in the question. So as I said before, let's take number 42, if I can remember rightly, osmosis in biology. Okay, osmosis, the movement of water. That's the question. It's just a statement, and you have to write everything you know. Now, the key word would be osmosis in this case. So we would provide and give them a definition of osmosis. Can someone give me a definition of osmosis?
Yes, I would say, for example, okay, osmosis, it belongs to the passive transport system, so it doesn't require energy. And it happens also through our cells, so that's also how the cells get exchanges of nutrients or water, but actually no, but actually osmosis is only the movement of water through a semi-permeable membrane from lower concentration to higher concentration.
Excellent. Yeah. Very good definition. So it's very, very important to know your definitions. Even if in the question or the statement, it doesn't say define a nucleotide, but it has nucleotide. It has these key scientific or biological terms. Immediately you should think, okay, that's the first thing I should define. Okay, so define keywords. That's very, very important. Imagine if we have a question about nucleotides and it says, identify the structure of nucleotides. Can someone define a nucleotide? Could someone maybe give me an example of if they had to define the definition of a nucleotide?
I mean, I can let. I mean, nucleotide is part of RNA or DNA, and it is. I'm not sure if it's made of an ribose sugar, or just a sugar, and phosphate. And
Exactly.
yeah, it makes up the DNA and RNA, yeah, this of the cells.
Perfect, perfect. So that's also a very good definition. So as you said, yes, the nucleotide is comprised of 3 components. It's comprised of a phosphate sugar, it's comprised of a nucleotide base, and it's comprised of ribose sugar, nucleotide base, and one more
Phosphate group.
and
Three
phosphate.
I don't know if it's 3 phosphate group. I'm not sure.
Yes, it is 3 phosphate groups.
Yeah, yeah.
And the sugar, it does vary. If it's in RNA, it's a ribose sugar. If it's in DNA, it's a deoxyribose sugar. And so these are the 3 components of a nucleotide. So that would be the definition. Okay. So the main point to take away when structuring your answer is to define key terms. Very, very important. Another thing is using comparison words when outlining the differences and similarities. So when it says, can you please compare DNA and RNA? Now, the main keyword here would be compare, okay? So you would have to use words where you are looking at the differences and the similarities. It's very important to look at both differences and similarities, not just the differences. And comparison words would be whereas, on the other hand, however, just to highlight these differences. Also to draw diagrams if necessary. So in this case, because the exam is online, it's not in person, the diagram bullet point is not necessary in this case, but maybe just to visualize your own ideas. So if it helps you just to draw a nucleotide structure, so when you speak to the examiner, you can say, yes, this is my diagram, these are the 3 components, and then it will be easier to illustrate your thoughts, just as a visual aid to help you. Also, it's very important to give examples when relevant to the question. So, for example, in chemistry, I can't remember specifically what question, but for aldehydes and ketones. So you would, of course, mention that it's part of a different homologous series. They are comprised of the same functional group of a carbonyl group. And you would mention where the carbonyl group is located or situated in a ketone and where it's located in an aldehyde. And then it would be very good to also give an example. So for example, a aldehyde would be propanol, okay? And you could draw the structure just to help you. And maybe you can show your notes to the examiner as well in the camera. Or a ketone, you could draw propanone, okay? And another thing that I keep re-emphasizing, provide as much detail as you can to impress the examiner. But as much detail as you can within the 15 minutes. Don't be too hard on yourself, because remember you have to cover two topics. So one topic from one subject and another topic from another subject. So yeah, just keep that in mind. Okay, are there any questions so far? Anyone would like to ask me anything? Okie dokie. If you do, just raise your hand, just let me know. Okay, so the different types of exam questions that I have found in the Silesia mark scheme are, first of all, comparison questions. Compare RNA, DNA, compare a eukaryote and prokaryote. Just comparison questions. This is where you would highlight the similarities and the differences. Another type of question is a definition question. Define the structure of a nucleotide. Define osmosis. Now, for those type of questions, one must think, how am I supposed to write for, let's say, 5 or 7 minutes? just about osmosis, and I've already defined it. Well, again, as I mentioned in the other points, you would mention examples. So osmosis in a red blood cell, osmosis in a plant cell. You would use other terms like hypertonic, isotonic, hypotonic, the differences in osmotic pressure. So with definition questions, sometimes it's hard to think how to structure your answer, because after you've defined the key word, one thinks, okay, now what? But it's very important to give examples of this biological process in natural organisms. Okay, and then the third type of question is the structure and function. So this would be, for example, the structure and function of the lungs, or the structure and function of the heart. And we will be covering these three types of questions today. Okay, so what I've done, because we are limited on time, and there are so many questions, there's an abundance of questions in the mark scheme for Silesia, I've just taken like a few samples from different subjects. So from biology, from chemistry, and from physics. Now, I have taken all from biology. I took 3, and I took 2 from chemistry and only 1 for physics, because the most popular is biology and chemistry. But I am aware that there was one of the students that said to me he's more passionate in physics. So if you wish to choose physics, go ahead. It isn't my forte. My forte is more biology and chemistry. But I thought I would just focus on bio and chem. Okay, and physics. Okay, so if you have, let's say, the mark scheme out in front of you for today, feel free to add more jottings down or other, let's say, side notes from today's session. So let's focus on the first one I just randomly picked. So this is a comparison question, okay, question 17. So a comparison of eukaryotic and prokaryotic cells. Okay, so this is quite a simple question. Now, how would we structure this answer? So I've written a few bullet points down. So the first one would be identify the type of question. So it's a comparison question in this case. It's asking to compare a eukaryote and a prokaryote cell. Okay, we would use comparison words like whereas, however, on the other hand. We would outline both the similarities and the differences, not just the differences. Also the similarities is very important in this case. And we will use subheadings to structure our answer. So for example, how does the cell structure differ? Organelles, we know that eukaryotic cells, they are comprised of multiple membrane-bounded organelles, whereas prokaryotes, not really. Yes, prokaryotes are comprised of ribosomes, which are also membrane-bound, but that is the only, let's say, one type of membrane-bounded organelle that they are comprised of. But then there would be an underlying difference. Eukaryotic cells, they are comprised of ATS ribosomes, and prokaryotes, they are comprised of 70S ribosomes. So this is why it's important to try to structure your answer with subheadings. And then look at the similarities and differences. So these would be the subheadings that I would encourage you to follow. So in the mark scheme, I am aware that there was a table that was drawn up and it was comparing between prokaryotes and eukaryotes according to these headings. So we have the cell structure, we have the size, we have the genetic information. So how is the genetic information organized? Is it in a nucleus and nucleoli? Is it organized in a nucleoid? Then organelles, as I mentioned before, membrane-bounded organelles present in eukaryotes, prokaryotes, not so much, ribosome size, and organisms. This would be examples of prokaryotes and eukaryotes. So eukaryotes, plant cells, animal cells, and then prokaryotes, you would give another example, so protista, archaea. Okay, so there we go. Let's start with the prokaryotic cell. Can I have any, can someone please tell me, let's say, a definition of a prokaryotic cell? How would you differentiate between a prokaryote and a eukaryote? Just in one sentence, 1 or 2 sentences. Okay, so I can see raised hands. Yes, you can go ahead and share your answer. How to talk. How to talk. There we go. Okay. So you can go ahead if you would like to share. How would you differentiate a prokaryote and a eukaryote? Just in 1 or 2 sentences. Just looking at the structure. Yes, yes, you can go ahead. Hello? Prokaryotic cells don't have a nuclear membrane. Very good. Spot on. Any other differences? Okay, so for this question, what I would encourage you to do is, okay, I will just have a look here on the chat. There is someone else. It's only one cell rather than multiple cells. Exactly, spot on. Very good. Yes. Anything else? You can also write down your answers if you're shy to share your answers. Okay, so as I mentioned before for this question, I did emphasize that because it's a comparison question, we have to mention the similarities and the differences, okay? So what I would encourage is to touch on the similarities between eukaryotes and prokaryotes, okay? So all cells, both prokaryotes and eukaryotes, they share four components. Okay, I'll just have a look on the chat. Prokaryotes lack a nucleus, whereas eukaryotes have DNA. And do you know how the genetic information, it's also shown on the diagram, is organized in a prokaryotic cell? And how is it organized in a eukaryotic cell? Because prokaryotes, they do lack a nucleus, but the DNA is organized as a nucleoid, whereas eukaryotes, the DNA or genetic information is organized in a nucleus, and it has a nuclear envelope, a nucleoli, nucleolus. Prokaryotes have plasmids, exactly, but eukaryotes have a number of chromosomes. Very good, yes. And then another underlying difference would be that eukaryotic cells, they undergo mitosis and meiosis, and prokaryotic cells, they undergo cell conjugation instead. Okay, very good. Any other differences anyone else would like to add? You can raise your hand or you can share. Okay, so in regards to the similarities, I will touch on the similarities first, okay? So first, I would define what is a prokaryotic cell, what is a eukaryotic cell, and then I would talk about the similarities. What do they share? What do they have in common? So first of all, Prokaryotes, eukaryotes, they both have ribosomes, all right? They have ribosomes. Can someone please tell me the function of these ribosomes? What is the biological significance of ribosomes in a cell?
I think there are protein synthesis. I'm not sure.
Spot on. Protein
Okay.
synthesis. Exactly. Sorry, someone wrote on here. Exactly, spot on. You're both correct. Yes, to synthesize proteins, protein synthesis. And the difference between prokaryotes and eukaryotes, you would emphasize this later on in the difference section, is the size of the ribosome. But now we're focusing only on the similarities, so what they share in common. Then we have DNA, as someone mentioned, the genetic material of the cell. So they both are comprised of DNA, another similarity. Third one, they are comprised of cytoplasm, okay? Could someone tell me the function of the cytoplasm? You know, in biology, there is a structure and there is always a biological significance to it. There is always a function to it, you know? A structure is not just there just for the sake of it. It always has a biological importance. So it's very, very important to always emphasize the function of that component. So in this case, cytoplasm is that jelly-like substance in the cell, both found in a prokaryote and a eukaryote, and where a lot of enzymes are found, a lot of chemical reactions take place to support and suspend cell organelles. Exactly. So for example, in a eukaryotic cell, the membrane-bounded organelles are all floating around in the cytoplasm. You know, they're all surrounded by this jelly-like substance, which is where a lot of chemical reactions are taking place. We were on prokaryotic cells and we were talking about the similarities between eukaryotes and prokaryotes. And so the last point that I was making was that each of them, both prokaryotes and eukaryotes, are comprised of a plasma membrane, which is this outer covering, let's say, lining the cell. But prokaryotes, in addition, they are comprised also of the cell wall as well, which is, we will go into the nitty-gritties, which is comprised of a peptidoglycogen, okay? All right, so these are the similarities between prokaryotes and eukaryotes. Now let's go on to the specific differences between them, okay? So as I mentioned before, there are some subheadings that I would encourage you to use. So let me just go to the previous slide here. So cell structure. As one of the students said here correctly, a prokaryotic cell is unicellular. Okay, these are very simple single-celled organisms. And does everyone know what the term is compartmentalization? Does everyone know what this term means? Just have a look here. Compartmentalization. This is basically the ability of a cell to compartmentalize itself. So prokaryotes, they don't have this ability, okay? They're unicellular and they are not comprised of membrane-bounded organelles. Whereas eukaryotic cells, they are capable of compartmentalization. So they divide themselves in little compartments. And this is the function of each membrane-bounded organelle. They are all divided from each other by a cell membrane. And this ensures that all the chemical reactions that take place, for example, within mitochondria or within lysosomes or within ribosomes, doesn't interfere with the other chemical reactions taking place in another membrane-bounded organelle. So one thing which is important to remember is that prokaryotes, they are not capable of carrying out compartmentalization. They are not compartmentalized. They're unicellular. They're simple cellular. Whereas eukaryotic cells, on the other hand, they are multicellular. Okay, they can also be unicellular as well, because an example are protista, which are unicellular. But animals and plants, they are multicellular. So what we're going to do is we're going to change from one slide to another. So the other subheading that we would cover is the size, okay? Another outlining difference. So the size of prokaryotic cells, they are usually extremely small. Does anyone know the size of a prokaryotic cell in general? Does anyone know in micrometers? What I will do is mute all of this so everyone is able to talk. Okay, this is just an additional piece of information. So for prokaryotic cells, they are 0.1 to 5.0 micrometers. Okay, let's see in the chat quickly. Yes, exactly. Yes, perfect. 0.1 to 5.0. Very, very important to put the units, micrometers. And for eukaryotic cells, they're extremely large. So they are 10 to 100 because they are packed with multiple number of organelles, membrane-bounded organelles, as we can see here in the picture. So the cell has to be much larger to accommodate, you know, all these organelles undergoing all their independent reactions. Then we go on to the genetic information. So how is the genetic information organized? And as one of the students correctly mentioned before, prokaryotic cells, the genetic information is organized in plasmids, which are circular pieces of DNA, which are found in the cytoplasm, floating in the cytoplasm. And there is also nucleoid as well. So the genetic information is arranged as a nucleoid structure. There's no nuclear envelope, there's no nucleus, there's no nucleolus, it's just a nucleoid. Whereas in eukaryotic cells, the DNA is arranged or found in a nucleus, and it's comprised of as you can see here, a nuclear envelope. Within the nucleus, there's a nucleolus. We've got chromosomes, as someone said correctly before. And a good difference to outline with the genetic information is that in prokaryotic cells, the DNA is circular in the form of plasmids. But in eukaryotic cells, the DNA is arranged as linear, okay? So linear in eukaryotic cells, prokaryotic cells, they're circular. Okay, then organelles. So in prokaryotic cells, as I mentioned before, there are not, the majority of membrane-bounded organelles are absent. The reason I say majority is because there is an exception here. There are these ribosomes, okay? And they are, do not forget, they are a membrane-bounded organelle, but that's the only organelle that prokaryotic cells consist of. And as one of the students said correctly, it carries out protein synthesis. On the other hand, in eukaryotic multicellular large cells, they're comprised of an abundance of membrane-bounded organelles, okay? So remember that in eukaryotic cells, we have animal cells, we also have plant cells, and there is a difference in structure as well. So, you know, eukaryotic cells further divide, and there are differences between the two. But can someone, I know it's mentioned on the diagram, but could someone point out some organelles that they know and tell me just briefly the function of them? Yes, you can just go ahead, you can raise your hand. So what would you find in a eukaryotic cell? An example of a membrane-bounded organelle and its function. So before we mentioned ribosomes, can anyone point out another membrane-bounded organelle that is found primarily in eukaryotic cells, both in plant and animal? Yeah, I'll just see on the chat. Perfect. Yes, spot on. Mitochondria is a powerhouse of the cell. Exactly. And it's responsible for ATP production. Very good answer. Yes. So when talking about that eukaryotic cells, they are comprised or they contain a number of organelles. Rather than just leaving the sentence like that, you would list the number of organelles that they are comprised of. And this is what we're doing now. So you would say mitochondria, which are the powerhouses. They are like these little energy, as one of the students said, these little energy powerhouses fuel, they fuel the cell. And the main energy source is ATP. Okay, you wouldn't have to go in too much detail on how ATP is produced within the mitochondria. You wouldn't have to go in too much detail because remember the time is running out. You've got 2 questions to answer, but it would be sufficient enough to say that the mitochondria are the powerhouses of the cell. They're important in active transport, hence the production of ATP, which fuels the cell, and for a number of reactions to take place. Okay, that's a very good one. Can someone else give me an example of another organelle and function? Just okay. So you can specifically mention it's a site of aerobic respiration. Anyone else would like to share? Okay, let's take endoplasmic reticulum. So we know that there are 2 types. There is smooth endoplasmic reticulum and there is rough endoplasmic reticulum. Can you please tell me, someone can raise their hand or write on the chat, whichever is easiest. What is the difference between smooth endoplasmic reticulum and rough endoplasmic reticulum in their structure? Go ahead, Federico. If you have something to share. I can't hear you if you are speaking.
Do you hear me?
Yes, I hear you.
Perfect. Okay. I think the difference is that in the rough endoplasmatic reticulum, we have ribosomes attached on, and smooth endoplasmatic reticulum not. So, so that's why I would say that in the rough, protein synthesis happens, and in smooth not.
Exactly, spot on. Yeah, that's the main difference. And also, as Aditi mentioned, rough endoplasmic reticulum, they have these ribosomes attached. And we know ribosomes, they are the site of protein synthesis. And for the smooth, they lack. There are no ribosomes. There is the absence of ribosomes. And does anyone know the function of the smooth endoplasmic reticulum? What is the main function of the smooth? So I can help you out here. The main function of the smooth endoplasmic reticulum is to transport materials, okay, between the organelles, primarily lipids, okay? So for the transportation between organelles of lipids, whereas the rough endoplasmic reticulum is for the transportation of proteins and protein synthesis, because they have these ribosomes attached to its surface. It's almost like a studded surface, whereas the smooth, as the name indicates, is very smooth. Okay. Okay, no problem, Federica. Right, okay. Then we have, could someone tell me Golgi body? The function of the Golgi body. The Golgi body can also be referred to as the Golgi apparatus. So either term is correct. Could anyone tell me, yes, what's the importance of the Golgi body in eukaryotic cells, plants and animals? So it's important to remember when speaking about these membrane-bounded organelles that you always talk about the structure, how are they arranged structurally, and then how does that relate to their function. So for example, when we spoke about the endoplasmic reticulum, I would say endoplasmic reticulum is in two forms, smooth and rough. The smooth differs to the rough, as we mentioned before. The rough carries out this function, the smooth carries out this function. In regards to the mitochondria, yes, we mentioned that they're powerhouses of the cell, but before mentioning that, I would also state the structure of it. So it's comprised of a double membrane, also is comprised of a mitochondrial matrix as well, inner and outer mitochondrial membrane. In regards to the Golgi apparatus, the structure, I would mention that it is basically an assembly of vesicles and folded membranes located near the cell membrane and stacked on top of one another. And as the DT says, sorting, modifying, packaging proteins and lipids. Exactly. Very, very good. Spot on. That would be a very, very good answer. And then you would just move on to the next organelle. Okay, so before I go down, okay, anything else that we can mention here? So the importance of the cell membrane. Does anyone know the importance of the cell membrane? Both in prokaryotic and eukaryotic cells. But remember that prokaryotes, they also are comprised of a cell wall. But in eukaryotes, do you know the importance of the cell membrane? Why it is important?
Yes, so for example, if they have a membrane, so it's membrane, first of all, it's, as we said, for example, in osmosis and all this stuff, it's a semi-permeable membrane, so he can allow some water and soluble things to pass through the membrane, whereas bigger macromolecules cannot pass through. And it is also kind of recognition and protection for the cell.
Excellent.
Whereas, for example, plants have okay, we're speaking about cell wall, hasn't cell wall, don't have, and these have also some benefits, pro and contrast.
Exactly. Yeah, so this plasma membrane, as you mentioned, it's semi-permeable and it's a selective barrier surrounding the cells. So it basically is controlling what substances are entering and what substances are exiting the cell. And if we go at a real, now we're going to the nitty-gritty, the details. If we go into the microscopic level of a plasma membrane, we know that the structure is this phospholipid bilayer, right? So we know it's comprised of phospholipids, which are comprised of a hydrophilic head and hydrophobic tail. We know that it's comprised of integral or intrinsic proteins, glycoprotein, cholesterol, extrinsic or peripheral proteins. These are all the components that are making up this plasma membrane. Anyway, for this question, I would just mention it's comprised of a plasma membrane and state its function. Okay, before we move on to the structures in the prokaryote, let's also cover, yes, lysosomes. Lysosome is another organelle which is important in the eukaryotic cell, both found in plant cells and animal cells. So does anyone know the function of lysosomes? They have a very important function. Have a look if anyone else has anything to share. Okay, I'll help you out. Lysosomes, okay. Right, let's have a look. Someone. Vesicles with hydrolytic enzymes, exactly. And they carry out the function of hydrolyzing. Okay, so they break down, or the hydrolysis of macromolecules into micromolecules or into smaller molecules or into remnants. Okay, so lysosomes are extremely important because they are these little vesicles packed with hydrolytic enzymes, which is needed for the degradation or hydrolysis. Use specific, well, when describing these processes or when describing a structure or membrane-bounded organelle, use terms like hydrolysis, degradation, biological terms. You know, you're trying to impress the examiner here, okay? Because we're going to become medics, we're going to become dentists at the end of the day. So we need to use scientific vocabulary. Okay, and let's have a look. Anything else?
Thank you.
Okay, peroxisomes. This is a specific organelle. Peroxisomes. Could anyone share with me anything?
There are, I think, there are responsible for the digestion of the cell. So, for example, also detoxification, because we also know it from the chemical. Peroxide is like I said before, to detox something. So it is like cleaning out the cell from things that it has to get rid of.
Exactly, spot on. And does anyone know, or do you know, Federico, the substance? Like, could you give me an example of a substance that they try to break down?
I think so. From the word peroxisome, I think it's peroxide. But
exactly, hydrogen peroxide.
Yeah,
exactly.
peroxide. Sorry, for the pronunciation. And
yes. So as you correctly said, they catalyze and they break down toxic substances. So they prevent the accumulation of these toxic substances within the cell. So they break them down. So again, if we talk about the structure of it, it's this little sac which is comprised of a number of these catabolic enzymes which break down these toxic substances, such as, always give an example, such as hydrogen peroxide. Okay, excellent. So these are basically, we call them universal organelles, okay, which will be found in a eukaryotic cell. You could go into further detail, you know, again, if you wish to talk about the additional organelles which are found in a plant cell. And could someone tell me very, very quickly, what other organelles would you find in a plant cell that wouldn't be found in an animal cell?
So I think chlorophyll, I'm not sure, chlorophyll for the photosynthesis, and the cell wall, for example. So
exactly.
Plants have a cell wall, whereas humans, for example, though animal cells don't have.
Yes, so as
and vacuoles also.
vacuole.
Yeah.
Mm-hmm. Exactly. So as you stated, a vacuole, which is usually large and central. We have a cell wall, okay? And we also have chloroplasts, yes. Chloroplasts, which are comprised of chlorophyll, which is that pigment, that chlorophylltic pigment, which is responsible for, anyone know the process? Very, very easy question.
Is it not a photosynthesis or pleural respiration? Or...
Exactly, photosynthesis.
Yeah.
Could we mention, yes, as Aditi says, could we mention cellulose too? Exactly, you could. So the cell wall is basically this external outer covering, which is comprised of cellulose, okay? And again, it provides support, mechanical strength of the plant. And then as you mentioned, yes, this vacuole. It's this fluid-filled, cavity, and it's important in trying to maintain the hydrostatic pressure, okay? But yes, as Aditi, as Federico correctly said, vacuole, chloroplast, and cell wall. These are the traditional organelles which are found in a plant cell. Okay, so now if we go back to the prokaryotic cell, okay, yeah, so if we go back, there are additional structures that are found in a prokaryotic cell, okay? Because remember, there are no membrane-bounded organelles except from the 70S ribosomes, but we've got structures which are important for prokaryotic cells to undergo, let's say, cellular conjugation, okay? Because remember, they're unicellular. So in this case, we have a cell wall. Remember, it's very, very different to the cell wall which is found in a plant cell, okay? We have a cellular capsule. We have pili, okay? Pilus is singular, but pili is in plural, or they're also referred to as fimbriae as well. We have these 70S ribosomes, as we mentioned before. They are important in protein synthesis. Now, let's say the components in a prokaryotic cell, they can be divided both into non-essential components and essential components, okay? So now this is going into further detail, trying to impress the examiner. So first of all, the essential components of a prokaryotic cell will be the cell wall, all right? So the cell wall, as I mentioned before, is comprised of this carbohydrate, this peptidoglycogen, which does differ between gamma positive and gamma negative prokaryotes, but that's going into a little too much detail. I would just mention that it's comprised of a cell wall, which is consisting of a peptidoglycogen, okay, which helps to keep the shape of the prokaryotic cell and prevent dehydration. Then another essential component of a prokaryotic cell would be a nucleoid, okay? We mentioned before, this is how the genetic information is arranged here. And it is circular as well, also found within the structure of a plasmid. We also have, it's not found, I don't think it's shown on the diagram, but we also have a cytoplasmic membrane as well in prokaryotic cells. And then another essential component would be ribosomes, okay? Does anyone know the two subunits that prokaryotic cell ribosome is comprised of? So we know in general that they're 70S subunit, but does anyone know the two subunits of a prokaryotic cell?
It's going to be dark. I'm going to try to do some dark and light. Not everything together because the grey, white,
Okay, it would be a 50 and 30S subunit. It's also sufficient just to say a 70S ribosome as well. Then the non-essential components would be the capsule.
I'm his weight stuff on there. I'm gonna do
Then the pili and the fimbriae. So does anyone know the exact function of a pilus or of pili in a prokaryotic cell? Why do these prokaryotic cells, especially bacteria, why do they have these little attachments coming from their surface? Have a look here.
So I think it's for distinguishing other cells, and because pilus or pili, they are no motile, so I mean they're just like there, and I think it's just the main thing, just to distinguish other cells and to attach them.
Mm-hmm. Exactly. So it's, as you correctly said, Federico, they are used to attach to other cells, other bacteria cells. Sorry, just before I continue, can everyone hear me well? I know some people are struggling to hear me. Is it okay now? Is the sound all right? Yes?
I think it's just like another somebody else who.
Is it better now? Slightly clearer? Maybe if I speak a bit louder. We can hear you fine, but there's a background noise. Okay. Yes, I can hear a noise too.
It comes from Ale.
I can hear also a background noise. Yes, it's coming from Ale. Just gonna, there we go. That's fine, and then I can unmute you afterwards. Can you hear, is that slightly better? Yeah? Can you hear me slightly better? Oh, perfect. Okay. So yes, as Federico correctly said, that these fimbriae or these pili, they are like attachments from the surface, and it's important for attaching to other bacterial cells or other prokaryotes. And also it's important for the exchange of genetic material. So if you say that, the examiner may say, okay, how do they exchange the genetic material and what process? So you can add to your answer conjugation. Okay, this is how prokaryotic cells, how they exchange the material through the pili. Okay. And they're basically described as these hair-like structures. Now, if you really want to go into detail, there are two types of pili. Okay, so there are ordinary pili and there are sex pili. So these ordinary pili, they play a role in bacterial adherence, okay, so to mucosal surfaces. Then you have these sex pili, which actually bind to other bacteria and are a tube for the transfer of large segments of bacterial chromosomes between other bacteria, okay? So you can just mention pili in general, or you can mention that there are two types of pili, ordinary pili or sex pili, which are also referred to as F pili. Okay, there's just a question here. Could they interrupt us with follow-up questions during the 15 minutes or wait for us to finish? Yes, they can interrupt you. So for example, in your answer, if you have been a little bit vague, so let's say if you have spoken about or membrane-bounded organelle of a mitochondria, and you've just said a eukaryotic cell has a mitochondria, they may interrupt you and say, for example, Federico, what is the function of the mitochondria? Okay, so they may ask you. So this is why it's really, really important when in that 15 minutes to put as much detail as possible, showcase to the examiner that you know what you're talking about. So this basically means that they won't have to interrupt you as often. But if your answer lacks clarity or if it lacks detail, they'll probably ask you some follow-up questions. So just try to put as much detail as you can to start with. Or if you have mentioned, let's say, a specific process, like cellular conjugation, they may say to you, or can you tell me about that? But that's going into further detail. But it depends, really. It depends how you structure your answer and what terms you use. Okay. Okay. And then last but not least, we have the flagella. So the flagella, again, is another very important component of a prokaryotic cell. And this is for locomotion. Locomotion, it allows the cell to swim, and for chemotaxis to take place, which is basically allowing it to swim towards food and away from poisons. You know, it's sort of like a mechanism. So this is really important. And according to the distribution of the flagella, there are 3 types. Again, going into further detail, there's the monotrichous flagella, which is only one flagella is present in the prokaryotic cell. For example, this picture shown here on the presentation, this is regarded as monotrichous flagella. We've got polar flagella, which is located in only one area of the cell. And we've got petrotrichous flagella, which is located over the entire surface. So you would have more than one flagella on the surface of the prokaryotic cell. Okay. Rightios. So I think we have covered the main differences, the main similarities. Always remember, when it's a comparison question, always specify differences and similarities. There are some questions in the mark scheme that just outline the differences, then just outline the differences. But when it says compare, okay, and it's a very generalized question, always specify similarities and differences. So this was, I think, mentioned or included in the mark scheme that was created by a group of meddling students, including myself. So this table almost summarizes what we've explained here. We have gone into much more detail about the organelles, and I do recommend you to go into detail about the structure and the function of each organelle. Remember, how is the structure adapted to its function? Very, very, very important to remember. Okay. So now we're still on biology. I am looking at the time. I am running over. Is everyone okay with time? I know here we're 9:45, but in England it's 7:45. Everyone okay with the time? Then just raise your hand or maybe write in the chat. You should be okay. All right. So this is another type of question. Okay. To raised hand. Yes, would you like to speak? I can also unmute you as well.
Oh, no, it's nothing.
Oh, are you sure? Okay.
Yeah, okay.
Okay, no problem. Okay, so this is another type of question, all right? A structure and function of the heart. So if we remember from the beginning, I outlined 3 types of questions that can be asked. A comparison question between 2 components like RNA, DNA, between eukaryotes, prokaryotes. I mentioned a definition question, for example, define osmosis, the movement of water. And I mentioned a 3rd type of question that can be mentioned is a structure and function, okay? And so I chose question 59. I thought this would be a good one to touch on today. Quite a detailed answer is required because there's a lot to cover. I remember one of the students mentioned to me, how much detail should we go into? And I think that's an excellent question. So let's see, how do we approach this question? Let's see about the heart. So we would identify the type of question. It's a structure and a function. How is the structure adapted to the function? Okay. Then we would state which system the heart belongs to. So the circulatory, the cardiovascular system. Then we would mention where it is located. So in this part, we could mention the borders, which cavity is it found in, the surfaces of the heart. And then we can talk about the structure. So the structure of the heart, we can subdivide it into a gross structure. So the chambers, remember the 4 chambers, the atria, the ventricles, and the valves of the heart. And then at a microscopic level, which will be looking specifically at the heart wall. So the endocardium, which is the inner layer. Endo means inner. Myocardium, myo comes from muscle, myocardium. Yes, and the muscle layer is comprised of these intercalated discs. This is how the muscle cell is distinguished between other types of cells, other types of muscle cells like skeletal and smooth. And then we have the pericardium, which is also referred to as the epicardium, which is the outer layer. Then we would explain the function of the heart. Okay, someone has just raised their hand. Okay, would you like to speak? Shall I unmute you? 1 second. Allowed to talk. Okay. Right. So if you have any questions at this stage, you can just raise your hand or write on the chat or just tell me. And then you would explain the function of the heart, okay? And then you would also describe the vasculature of the heart as well. So the blood supply, the venous drainage, and this is how you would structure this type of question, really. This could be also applied for if we're talking about the structure and the function of the lungs, if we're talking about the structure and the function of the urinary bladder, you know, always mention which system is it part of, where is it located, the borders, the surfaces, describe the structure, okay, at a microscopic level as well as at a gross level, explain the function of it and also the vasculature as well. So also if it has innervation as well. Okay, so are there any questions at this stage that anyone would like to ask me? So I see this type of question almost like a journey. You're trying to bring the examiner on a journey, okay? So you start from very, very generalized point, okay? Which system is it part of? And then you go into the nitty-gritties, you go into the details of it, and you guide the examiner about the heart in this case, or about the lungs, okay? So I see it as taking the examiner on a little journey and also to showcase your knowledge. It's very important. Okay, so if we look at the heart structure grossly, could someone please tell me what are the chambers of the heart? What are the 4 chambers of the heart? You can feel free to write in the chat, or you can also unmute your microphone.
So if you want to, I can say we have the right atrium, right ventricle, left atrium, and left ventricle, and the 2 valves, so the tricuspid and the bicuspid, are also called mitral valve, are there to prevent backflow of the heart, of the blood. And so the structure is sorted out like that, just to prevent the backflow and to get less oxygenated blood into more oxygenated blood, so with more oxygen. And this function is just to pump blood into the body and to put oxygen into it. And
Exactly.
from the structural view, so the left side is a bit bigger than the right side of the heart.
And do you know why that is? Why is it slightly thicker on that side?
I think it is thicker because of the muscular work, perhaps, which it's needed to pump out then the blood into the body. So it needs, I think, a great work to do it. So that's why it has a bigger myocardium, the muscular layer. So I think because of that.
Exactly, spot on, Federico. Yes, exactly. So the left wall is slightly thicker because it's under a lot. It's working much harder. You know, it's withstanding a lot more pressure. So it's pumping and working much harder. So the muscle, of course, is undergoing hypertrophy. It's becoming thicker, and at the end, it's seen, let's say, grossly as a much thicker wall compared to the other side. Exactly. So as we know, and as Federico said nicely, that the heart is consisting of these 4 chambers. So we've got the 2 atria and we have the 2 ventricles. All right. So we have the blood, which is returning to the heart, enters the atria, and then is pumped into the ventricles. And from the left ventricle, blood is passing into the aorta and enters the systemic circulation. Remember, we have this dual circulation, systemic and pulmonary circulation. So from the left ventricle, blood is passing into the aorta and entering the systemic circulation. But from the right, it enters the pulmonary circulation, so via the pulmonary arteries. Okay. So as we mentioned, we've got the atria and the ventricles. We have the right atrium. We have the left atrium, which is very nicely illustrated here in this diagram. And we also have the ventricles too. So in the right atrium, this receives deoxygenated. Okay. Does anyone know where it's receiving deoxygenated blood from?
Yes, so I think here it is the exception. So normally we know that arteries have more oxygenated blood than veins, but in the pulmonary artery and veins, it is just exchange, is the opposite. So the pulmonary artery, so it gets enriched in the lungs. So we get from the lungs oxygen. So through the pulmonary veins, so we get oxygenated blood, and through the pulmonary artery it goes into the lungs, so less oxygenated blood. So that's the only exception about artery and veins, where it is less and more oxygenated.
Exactly. Very good observation. And yeah, so usually we know regarding arteries and veins, but in this case of the heart, as you mentioned, with those pulmonary arteries and with those pulmonary veins, it's slightly different in regards to the heart. So in regards to the right atrium, it's receiving the deoxygenated blood, okay? So from the superior and the inferior vena cava. And the left atrium is receiving oxygenated blood, okay? So from the 4 pulmonary veins, which are specifically shown here on this diagram. Okay, and then the ventricles as well. So the right ventricle, could you tell me, is it receiving the right ventricle? So is it receiving deoxygenated blood or is it receiving oxygenated blood from the right atrium?
So I can answer if you want to. It is still, because nobody's answering. So because it's still deoxygenated blood, because it has still to go through the pulmonary artery into the lungs. So it's still deoxygenated.
It's still deoxygenated. Excellent. Yes. And also, as shown in the diagram, the blood is blue, so it's deoxygenated still from the right atrium. And then you have the left ventricle, which is receiving, as it's shown here, oxygenated blood from the left atrium. So for the gross structure, I would mention specifically those four chambers. So I would mention, as Federico said, the right atrium, what type of blood is it receiving, the left atrium, as well as the right and left ventricles. And then we go on to the valves, okay? So we know that the valves, they are divided into, let's say, two types of categories, okay? Could you mention to me what type of valves we have? Okay, bicuspid and tricuspid. Very good. And what is the difference between the bicuspid and the tricuspid? Quite an easy question. It's also mentioned in the name as well. Yes, the tricuspid is divided into 3 leaflets and the bicuspid is divided into 2 leaflets. Exactly, spot on. And these are referred to as the atrioventricular valves, which are located between the atria and the ventricles. Okay. And then also the bicuspid valve is also referred to as, I think Federico said, also as the mitral valve as well. Then we have the semilunar valves. Okay, do you know where the semilunar valves are located? So the atrioventricular valves, they are located between the atria and the ventricles, also as the name indicates and suggests. And so their function primarily is to close during the start of ventricular contraction, which in this case is systole. Okay, remember we have systole and diastole. So diastole during relaxation and systole during contraction. So the atrioventricular valves, they close during the start of the ventricular contraction. Whereas the semilunar valves, do we know where they're located? Can anyone mention where they are located? Have a look, there's an answer on the chat.
Pulmonary artery and vein.
Exactly. So between the aorta and the left ventricle. Exactly. And so these semilunar valves, we have 2 types. Okay, we have the pulmonary valve and we have the aortic valve. All right. And can someone tell me what is the function of the semilunar valves? So we mentioned the function of the atrioventricular valves. What is the function of the semilunar valves in this case? Prevent reflux of blood. Yes, exactly. Preventing the backflow of blood. So ensuring the flow of blood is in one direction. So basically they close. So rather than opening, okay, they close at the beginning of ventricular relaxation, so diastole. So again, try to use these specific words like diastole, systole. So just a quick recap, the semilunar valves, they close at the beginning of ventricular relaxation, so diastole. In comparison to the atrioventricular valves, which close during the start of ventricular contraction or systole. Okay, so semilunar valves, we know they're divided into pulmonary and aortic valves. Okay, so we have mentioned that. Now we would go on. So this is, of course, the gross structure of the heart, and we have the microscopic structure. Okay, so the three layers, as I briefly mentioned before. So could someone briefly explain to me, yes, the three layers of the heart. So I would encourage you, when talking about the microscopic structure, when you specify the three layers, mention where they are located. Okay. Mention where they are located, what cells they are comprised of. Okay, so as I mentioned before, the myocardium is that middle layer, it's the muscle layer, and it's comprised of these intercalated discs. We know the heart has its own electrocardial system with these Purkinje fibers. You can go into detail regarding the electrochemical system or electrical conducting system if you have time within the exam, but that would just be some extra detail. Then you've got this endocardium, which is the inner layer, and then you have the pericardium. And there are two types of pericardium. There's the parietal and there's the visceral. So the visceral is in contact with organs, and the parietal is in contact with the skin. Okay. So now if we look at, then one of the points for this answer was the function of the heart. Can we outline some functions of the heart? You can write them down on the chat. So we know that the heart is part of the cardiovascular system, the circulatory system. So what is the function of the heart? We already mentioned this, but just to re-emphasize it. Anyone would like to share the function of the heart? To pump blood around the body. You know, it's acting as this pump to pump blood around the body, oxygenated blood to all our tissues in the body. Okay, let's swiftly move on because I'm seeing that my technical glitch took up some time. So the vasculature of the heart. So we've got the blood supply and we've also got the venous drainage. So in this case, I would talk about the main the main arteries. Okay, so the coronary arteries in this case. I would talk about the pulmonary arteries. I would talk about the aorta, okay, and their function as well. For example, in case of the aorta, you could talk about the branches of the aorta. You could mention that it's the largest artery in the body, mention its function. Then you can talk about the coronary arteries as well, the left and the right one. These are just, you can see here, left marginal artery, left anterior descending artery. These are just additional branches. Just try to identify the main vessels, okay, and their function. And we know the function of the arteries. And then we have the venous drainage. Okay, it's important again here to mention the main veins of the heart. So we have the superior vena cava, we have the inferior vena cava as well. Okay, you could also mention the aortic sinuses, the great cardiac veins, the small cardiac veins. But again, just try to, because there's just so much information, just try to structure your answer again. So when you talk about the vasculature, when you talk about the venous drainage, about the blood supply, just mention specifically the main tributaries, in this case, when we're talking about venous drainage, or the main vessels involved in blood supply, their function, what type of blood are they carrying, deoxygenated or oxygenated, and then just move on, you know, keep on going with your answer. And then also, once you've written your answer, you can go back if you have time at the end and add more and extra details in as well. Okay, and so now we move on to chemistry. Are there any questions in regards to biology? Because, you know, we don't have the whole evening, we just have a specific time frame for tonight because it's only a taster session. I just picked out 2 types of biology questions, which I mentioned in the mark scheme, and just gave an overview of how one could structure the answer. Are there any questions so far? Okay, I didn't see any questions. Okay, so chemistry. Right, so in chemistry, I chose again a comparison question, differences between aldehydes and ketones. So just an overview of how I would structure this question. It's a comparison question. And this is a classic example of it being a comparison question, but it's only highlighting to mention the differences, okay? It doesn't highlight the similarities, it just says the differences. So in this case, just answer specifically what they would like. Define the key terms, so what an aldehyde is and what a ketone is, okay? Could someone do that for me now? Can someone please tell me, yes, what is an aldehyde and what is a ketone? Can you tell me specifically what functional group they contain? You can also write it down if you wish. What functional group does the aldehyde and ketone contain? Sorry, can you repeat that again?
The carbon functional group still.
Exactly. Yes, carbonyl group. That is the functional group which is shared both in aldehydes and ketones. Then we could also draw the structure of a ketone and an aldehyde, okay, to illustrate the location, because the difference between them is the location of this carbonyl group. In aldehydes, where is the carbonyl group found in aldehydes? Is it found at the end of the chain, or is it found within the chain, within the carbon chain, or within the carbon backbone? Where is the carbonyl group located in an aldehyde? Let's have a look. Okay. All right. So in regards to an aldehyde, we have a few answers. So within the carbon chain, within the backbone, YM says at the end of the chain. Okay. So in regards to an aldehyde, YM is correct. The carbonyl group is found at the end of the chain, okay? Whereas for a ketone, where is the carbonyl group found? It will be found within the carbon chain, in the middle of the chain, exactly. Perfect, spot on, okay? So this is really important to remember. So an aldehyde and a ketone, yes, they are part of different homologous series, but they do share the same functional group. An aldehyde is comprised of a carbonyl group, which is at the end of the carbon chain, and a ketone is comprised of a carbonyl group, which is found in the middle of the chain. Okay, and this the location of this carbonyl group affects how they are named as well. Okay, and we'll come on to this. So you can also draw the structure of it just to try to illustrate your answer, also to hold it up to the examiner to show that you've understood where the carbonyl group is located. Also to describe which tests can be used to distinguish between an aldehyde and a ketone. So the Benedict solution, what other tests are there? We've got the acidified potassium dichromate solution. We have the tolerance reagent. So these are all tests to basically distinguish between an aldehyde and a ketone. And then you can also describe the properties of the ketones and the aldehydes as well. Because the location of this carbonyl group primarily affects also their properties. So like the boiling points, the melting points, their volatility. Okay, so this is the structure of an aldehyde. Okay, so this is the double bond attached to the carbon and the oxygen. This is the carbonyl group. We have R. Does anyone know what R represents? So what does R stand for? Okay, R stands for the variable group. Okay, so it can be this other carbon can be bonded to another carbon, which can then be attached to another double bond oxygen, to an alcohol, an OH group, to another functional group. So this variable is what makes aldehydes, so different, because they can be containing other functional groups as well, or they can be attached to other carbons. So as we elongate the carbon chain, of course, this will influence how we name the aldehydes as well. But the suffix of an aldehyde is -al, anal. Okay, this is very important. And always to remember that this carbonyl group is found at the end of the chain. So if we would have an aldehyde which is comprised of 3 carbons, how would you name an aldehyde which is comprised of 3 carbons? Unfortunately, I can't annotate it. So if you had 3 carbons, a 3-carbon aldehyde, how would you name it? Anyone? Okay, so a chain of 3 carbons will be propanal. Propanal, okay? And so when talking about the structure, let's say, of an aldehyde, I would always give an example. So draw the structure of propanal, draw the structure of butanal, pentanal. Yes, anything that is showing the structure of the carbonyl group at the end of the chain, but varying number of carbons. Okay, so this is the aldehyde, comparing it to the ketone structure. So here we can see that we've got 2 variable groups on either side. So as Wayam correctly said, that this carbonyl group is found within the carbon backbone, in the middle of the chain. And this influences how we name a ketone. So in this case, if there were three carbons, we would name the ketone as propanone. And you could also specify where the carbonyl group is found. Okay, so if it is found on the 2nd carbon, it could be propanone 2-one. All right. Any questions so far that you would like to ask me? Okay, so moving swiftly on to the different tests that can be used to distinguish between a ketone structure and an aldehyde structure. We've got a few tests. So for example, we have the two famous ones, or three rather. We have the Tollens reagent. Okay? We have the Fellings solution as well, and we have the Benedict solution. So I think this is really, really important after mentioning the difference between the structure of a ketone and an aldehyde to also specify the different tests to distinguish the two structures. This will showcase to the, to the examiner that if you were to do, let's say, a biochemical test, you would be able to identify, is it a ketone in there or is it an aldehyde, okay, due to the final result after using a specific solution. So for example, with Benedict solution, okay, for a ketone, there will be no change. So the Benedict solution is blue to start with, and there will be no change in the blue solution. But for an aldehyde, the blue solution would produce a dark red precipitate, okay, of copper oxide. So if you want to distinguish between a aldehyde and a ketone and you use Benedict solution and there's no change, we're dealing with a ketone. Okay, remember the carbonyl group is in the middle of the chain, it's within the carbon chain. But if there is a dark red precipitate which is formed, we know an aldehyde is formed, okay, or we're dealing with an aldehyde. The second solution would be Fellings solution or Fellings reagent, okay? And in regards to this, a ketone, again, there would be no change in the blue solution. But for an aldehyde, the blue solution would produce, again, another dark red precipitate as well. Now, what you could do is you could also write the equation of the solution. This would also impress the examiners as well. So in the mark scheme, the equation of, let's say, the complex which is formed is written in there. So I would learn the formula of what Fellings solution is made up of what Benedict solution is made up of. And then we have Tollens reagent. So Tollens reagent, again, if we're dealing with a ketone, no change in the colorless solution, okay? Whereas for the aldehyde, it would produce a gray precipitate, okay? This Tollens reagent is also referred to as the silver mirror test, because if you're dealing with an aldehyde, the end product is like a silver mirror because a silver precipitate is formed. So you can literally, if you hold the test tube in front of you, you can literally see yourself in the test tube. And this would indicate that you're actually dealing with an aldehyde, because the aldehyde itself is to a salt of the corresponding carboxylic acid. So again, when talking about these different tests to distinguish between an aldehyde and a ketone, I would write down the formulas of what is formed at the end and what changes would be seen if there was an aldehyde and what changes would be seen if there was a ketone, and that would suffice. That would be fine. Okay, moving swiftly on. Question 52. I thought to choose a structure question now. Okay, so as you can see here, it just says the structure of nucleotides is very, very vague, very, very vague, and one could write a lot about nucleotides. Or one could write very little about nucleotides and just write the definition. But here it's talking about the structure. So you would speak about the three different components of the nucleotide, the function of it, the difference between DNA and RNA. So what would we cover in this type of question? So we would identify what type of question it is. So in this case, it is a structure question. It isn't a comparison question, it's just a structure question. We will define the key terms. So in this case is a nucleotide. So as we mentioned before, we mentioned the 3 components. When we're talking about the definition, we need to specifically mention what is it. Okay, what is a nucleotide? All right. Then we would talk about the structure. Okay, and whenever we talk about the structure or the components, we always talk about the function. Okay, how is it adapted for its function? And then we could at the end highlight the differences of nucleotides found in RNA and in DNA as well. Okay. So the structure of the nucleotide, as we mentioned before, 3 main components. So we have a phosphate group, yeah, as we can see here. We have PO4 3-. We have a ribose sugar, and we have a nitrogen base. Okay, so these are the 3 components which are found within a nucleotide, and they all have different functions. As we specified before, there are 3 phosphate groups, and also the ribose sugar does differ. So if it's in RNA, it would be the ribose sugar. If it's in DNA, it would be the deoxyribose sugar. So in regards to the phosphate group, okay, this is a very, very important part of the molecule. And we know that ATP, we mentioned before that ATP, okay, is found within the mitochondria, and this ATP is this energy source, okay, it's this fuel, okay, and it is allowing all the different reactions to run. It's an active process, okay. So this ATP, this molecule, is this fuel, and it contains these 3 phosphate groups which can store a lot of energy in their bonds. So when this ATP molecule is broken, a lot of energy is released into the atmosphere. So we know that the bonds which are formed within a nucleotide, can you tell me what type of bond is formed within a nucleotide? So when, for example, 3 nucleotides are bonded together, the phosphate groups, they are attached to other phosphate groups by a specific bond. Could you tell me what type of bond that is? Does anyone know? What bond attaches each phosphate group of the nucleotide? A phosphodiester bond. Okay, so the phosphodiester bond binds each phosphate group of the nucleotide. Then we have the second component, which is the ribose sugar. And as I highlighted before, there is a difference between the sugar found in DNA and the sugar found in RNA. Okay, so for RNA, we have ribonucleic acid, whereas in DNA, it's a deoxyribonucleic acid. So in DNA, the sugar would be deoxyribose, whereas in RNA, it would be the ribose sugar. So the sugar has, let's say, an exposed oxygen, and this can bond with the phosphate group of the next molecule. And then we have the nitrogen base. So we have 4 main nitrogen bases, or they're also referred to as nitrogenous bases, and they can be divided into 2 groups. So they can be divided into purines, and they can be divided into pyrimidines. So can you please tell me What is the difference between a purine and a pyrimidine in their structure? Because these 4 bases, some of them are pyrimidines, but some of them are purines. Could you tell me what is the difference between a purine and a pyrimidine? You can also write on the chat or can unmute you. Mm. So the difference in structure between a pyrimidine and a purine. Okay, should I choose someone at random? Federico, can I ask you?
Yes.
You've been very active during this session. Can I ask you, what is the difference? What is the difference between a purine and a pyrimidine? Just very simple terms in structure.
Well, I have to be honest. Okay, so the question was between, you said, I don't know, to be honest.
Okay, no problem, no problem. Okay, I'll help you out in this one. I'll help you out. So purine, okay. So as I mentioned, we've got 3 components of a nucleotide. And the nitrogen bases are divided into two types of groups. We've got purine and we've got pyrimidines. So purines, they exhibit a double ringed structure, double ring. But a pyrimidine exhibits only 1 ring structure. Okay, so we've got on the chat here. Let me have a look. Exactly, so purines are structured as double carbon nitrogen ring with 4 nitrogen atoms, while pyrimidine is a single carbon nitrogen ring with 2 nitrogen atoms. Well said. Very well put. Very well. Yeah, so that's perfect. So that is the difference between the pyrimidines and the purines. And then I would mention which, let's see, which bases are pyrimidines and which bases are purines. So for the purines, which exhibit a double ringed structure, would be adenine and guanine, okay? And for a pyrimidine, we would have, it exhibits only 1 ring structure and it would contain cytosine and thymine. And this is in DNA, this is found in DNA, but in RNA, there is 1 base which is different and replaced by another one. Can someone tell me what base is not found in RNA? It's replaced by something else. Okay, so I'll say that in RNA, thymine is not present and it's replaced by, can someone tell me? Okay.
Yes. So this, I know, this is one of the few things which I know from chemistry, because it's related to biology. So we have an RNA, we find uracil
Yes.
instead of thymine. And
Exactly.
in the DNA, we found adenine, guanine, cytosine, and the last one I don't remember. Thymine.
Cytosine, guanine, thymine, adenine found in DNA. And there's Federico, and let me see who said it. Rahul said correctly that yes, in RNA, no thymine is present, but uracil is present instead. And we know that there is this complementary base pairing, okay? So we have a purine and a pyrimidine base bonding together. So could you tell me which base, so let's find adenine. Adenine, which other base does it bond to? And which base does cytosine bond to, for example? And it's important to mention what type of bond as well. They are bonded by hydrogen bonds, hydrogen single bonds, okay? Or hydrogen bonds. So adenine nucleotides or adenine bases, they bond to which other base for adenine, using this theory of complementary base pairing theory.
So I can say, if you want to guanine, I don't know the pronunciation, guanine, so adenine to guanine, I think, and cytosine to thymine. I'm not sure about that, the replacement.
Okay. Really close. Okay, so I have a little tip. Okay, so we know that the purines are adenine and guanine, and we know that the pyrimidines are cytosine and thymine, and a purine and a pyrimidine, they bond together. Okay, in regards to this theory of complementary base pairing. So when we talk about complementary base pairing, the adenine binds to T nucleotides, okay? And the cytosine bonds to guanine. And the way I remember this is that C is like a curly C, it's a curly letter, and G is also, excuse me, G is also a curly letter as well, okay? So if you see it like that, this is why they are complementary base pairs, right? So you've got one pyridine, one purine, but to remember which one bonds to which, I just remember we've got curly C and we've got curly G, they bond together, and we've got straight A and straight T that bond together. I was always told that by my biology teacher at high school. Exactly Ryan. A to T, C to G. Always a pyridine and a purine, they always bond together in terms of hydrogen bonds and in terms of complementary base pairing. So when talking about let's say overall, in conclusion, when we're talking about the structure of a nucleotide, you can draw it out, you can show it to the examiner, you can mention the three components and always mention the function. Always mention the type of bonds. So the phosphate groups, they are bonded together by phosphodiester bonds, but the nitrogen bases, they are bonded together by hydrogen bonds. In terms of 2 hydrogen bonds or 3 hydrogen bonds. Sorry, I made a mistake before, depending if they're purines or pyrimidines. Okay. Right, and quickly, time 10:35, so 8:35. Now we'll look quickly at physics. Physics is not my forte, but we'll have a go. Physics, I choose quite an easy one, but the crystal structure. But the whole point of this taster session is to look at how to structure these answers, whatever question we come across, okay? So I chose just this one, a crystal structure, which could be seen as a definition question. Okay, so we're defining the key terms of solid state and crystal structure, because we know that there are three states of matter. There is liquid, solid, and gas. And crystal structure is a type of solid state. Okay, if I'm not mistaken. You know, in general, solids, they can be classified into the crystalline particles or into the crystalline structure, which are orderly arranged. They have a specific order. Or you've got those which are amorphous structures. So where the particles are randomly orientated rather than regularly arranged. And then you would describe the structure, okay, of a crystal structure, and then also identify the properties of it as well. So this is how we would structure this type of question. So this is just a brief diagram of what type of units are found within the crystal structure. Could someone give me a definition of crystal structure in physics terms? Okay, so remember with this type of question, where it's relevant, always mention the definition. Always define the key words. Okay, so we mentioned that in solid states, there are different states of matter, and in solid states, there are two types. They can be further divided into a crystal structure, so crystal particles, which are arranged in a sort of structured arrangement. And then you've got those which are amorphous, which are randomly orientated. Now, in regards to a crystal structure, could someone give me a definition? How would you explain it to the examiner? What are the components of a crystal structure? Feel free to, I think this is the last question, everyone. But feel free to tell me your thoughts of what a crystal structure is. How it? Okay, should I randomly pick someone? Waim. Wayam? Am I pronouncing that correctly? Could you give it a go? What do you think crystal structure is? They can also unmute themselves, right? Okay.
Hello?
Hello, hi.
Yes, hi. Yeah, what did you ask again? I didn't.
Oh, okay, no problem, Mayim. Okay, yes, I asked, could you give me a very brief, simple definition of a crystal structure?
I didn't have physics.
Oh, you didn't have physics?
Yeah, I haven't chose the physics either.
Mm-hmm.
Okay, so I just.
Is there any? Oh, no problem. I would have also chosen biology and chemistry, but are there any physics whiz in this group quickly that could tell me the structure or the definition of a crystal structure? Okay, we can just briefly describe it. So in terms of if someone chooses physics and this question randomly, we will talk about that a crystal structure is basically formed only when the group of atoms are arranged identically at the lattice point. So it can be summarized by using an equation such as the lattice and a basis, and this will form the crystal structure. So from what we can see here, what is shown, we can see primitive and non-primitive types. Okay, so this is the two types of unit cells. So a primitive cell is a minimum volume unit and has only one lattice point, and the non-primitive type has more than one. And then in regards to this crystal structure, you would talk about the properties of it, how I mentioned the two types of unit cells, the properties, the arrangement. But I would right at the beginning talk about the three states of matter, then talk about solid state, how is it further divided, amorphous and crystal, and then start talking about the crystal structure. But it's very, very important to start with the definition. Definition is very important. So that's something very, very short about crystal structure and about physics. But if you do choose physics, just apply what we have covered today about how to structure your answer. You know, identify, is it a comparison question? Is it a definition question? Is it a structure and function question? And use scientific terms. This will always impress the examiner. So in this case, lattice points, non-primitive type, primitive type, periodicity. So the properties of the crystal structure as well. So I have just gathered here a few recommended study resources that I would recommend you when preparing for these questions. So the mark scheme that we gave you, that team of medical students here at MedLink put together, but just remember that that is just as a basis, just as a foundation. A lot more detail can be added. So you can use these sessions if you wish to continue, and we can focus on specific questions that you would like to add more detail to, or you would like more guidance or more support in. Other resources such as Teach Me Anatomy is extremely very, very good for biology. I recommend, highly recommend. KenHub as well, but to access the other comprehensive videos, you have to become a member. But some free resources are good, but I recommend also BioNinja for biology, Chem Guide for chemistry, very good comprehensive notes there. But Teach Me Anatomy is very good for structure and function of organs, so such as the heart, such as the lungs. Yes, and I wish you all good luck. And does anyone have any questions? And I hope you enjoyed this evening. I'm sorry if I ran over time, and I do apologize for the technical glitch. But I hope maybe I will see some of you later in the during the Christmas time for the preparation of these sessions. Osman is coming along too.
Yeah,
My fellow colleague.
thanks for joining us, guys. It was. I hope it was helpful for you. Let us know what your thoughts are in an email. We'd really appreciate that. Your feedback is really important for us and for your other colleagues as well who are learning alongside you. So thanks all for joining. We loved that you were interacting with us and chatting with us and talking to us as well. We really appreciate that. Yes, this class is recorded. Again, if you have any questions that you need answers for, let us know. And we look forward to receiving your feedback through email. We'd really appreciate that if you let us know. And let us know as well if you'd be interested in signing up for these lessons so May can teach you. May is, as you see, a very good and studious student. She's the top of her class. She's fantastic at what she does, and she's this is not the first time she teaches, as you can see. She has lots of experience teaching and helping students like yourselves. So if you would like us also to cover specific topics in future, then let us know, and we're really excited to teach you guys and show you what you need to do and how to answer questions and how to be the top in this entrance exam so you can ace it and get into medical school and pursue your dream of studying medicine. All right, so have a good evening, everyone. It was lovely having you with us, and we look forward to the next time. All right.
Thank you.
Bye.
Okay, thank you everyone. Bye-bye. Look forward to hearing from you.